welcome to this afternoon session of the Clinam conference. It's going to be a joint session. It used to be an experiment which we have done last year for the first time, in the sense that we have a meeting of a German priority program called SPP 1313 and two national Swiss programs. I have been acting as the chairman of the German program, and for that reason I have the honor to open this afternoon session. I would like to tell you a few words about this German program. Thereafter, my two Swiss colleagues, Louis Schlappach and Peter Gehr, will say a few words about the Swiss national programs. The program that we have been running in Germany was called Biological Responses of Nanoparticles. The idea of this program was to investigate artificial nanoparticles in the presence of the biological environment, with an aim to understand how do particles transfer across membranes, how do particles enter cells, what are the different mechanisms, how they do it, how does this depend on the properties of the particles. It's a very, very important issue. You probably all know that biological responses of particles is related to a number of parameters: size, surface composition, shape, elemental material, and so forth. We know that there are particles that are almost inert in the biological environment, but we also know there are some of them which are really pose a huge risk to the biological systems if they are in their presence. The program we have been running in Germany, sponsored by Deutsche Forschungsgemeinschaft, has been running over 6 years. We have just been terminating this program. We are in the process now of writing it all together. And we have decided to make a final report in a special issue of the Beilstein Journal of Nanomaterials, which is going to accept all these papers, provided they all pass the reviewing system, and we will have this in publication probably towards the end of this year. Further, a few items that may be important. We had a number of about 37 groups working together. So it was essentially the whole German community of biologists, biochemical engineers, technical chemists, medical people, and cell biologists and what have you. And we have used a number of different techniques, advanced techniques, to do all the characterization of the particles and also the cellular systems. Today, we have 3 different colleagues invited to present this program to you in 3 different independent talks. And I would like to say a few words about these colleagues as well. The first speaker is Professor Katharina Landfester. She's sitting here in the first row. And she will be talking on nanocapsules and their specific interactions with cells. Frau Landfester is a director of the Max Planck Institute in Mainz of polymer chemistry. She has been there for the last 3 years, approximately. 4 years? 5 years? Oh, time is running very fast. It's usually the case, 5 years. And she, I believe, is the one who has been very active in designing and manufacturing polymeric systems, polymeric nanoparticles, also with the aim to not just enter cells, but also to, in order to carry drugs and other materials into certain environments in the biological system. So we look forward to her talk. The second person is Professor Eckhard Rühl. He's a physical chemist from the Free University of Berlin. And he is talking about the uptake of nanoparticles on the skin and into cell. In his program, which he has been running in Berlin, he has been very closely connected to another colleague who is kind of a skin specialist, if I may say so. He is not here today, but you have been involved with his work, and he is taking over in presentations on this subject as well. Regarding these 2 people, an important issue I would like to add, and that is that they have been able to initiate, each of you, one in Mainz and one in Berlin, a so-called Sonderforschungsbereich from DFG. What is this in English? Huh? collaborative research centers. I was talking about priority program, this is collaborative research centers in Germany. It's a very distinguished and very prestigious institution. Normally it means that people assemble at the same place. The units should be at the same place, and it's always a documentation of the strengths of a particular university or research establishment to be involved in these kind of programs. But anyway, what I was going to say, there have been 2 initiatives now, one in Mainz and one in Berlin, and both are supposed to continue the kind of work that we have been doing in the priority program which I was coordinating. The third speaker is Professor Stefan Bartschikowski. He's a technical chemist, but he is a chameleon in the sense that he also talks about cells, he talks about cellular interactions, but in principle he's a manufacturer of nanoparticles. And that's what he's talking about today. He talks about reproducts effects of alloy nanoparticles on reproduction, meaning that these particles have been formed and have been used in the program to investigate the effect of the reproductive system. It has been tried on animals and probably is going to show you important results from this kind of studies. So with this, I like to be closing my little introduction, and I hand the floor over to Louis Schlappach to introduce to you now the general vision of one of the Swiss programs.